In 1955, a Norwegian explorer descended into a volcanic crater and found a tree with a single living branch. The rest of the island had been cleared 2 years earlier, but this turned out to be only the beginning of the mystery. He gathered a handful of seeds from it and sent them to Sweden. 4 years later, 4 seedlings sprouted. The species seemed saved, but all 20 attempts to return the tree to the island ended the same way. It died. Only decades later did it become clear that something else had disappeared from the island along with the tree. The Toromiro, Sophora toromiro, is a small tree of the legume family. Twisted trunk, reddish-brown bark, yellow flowers. It grew exclusively on Easter Island, nowhere else in the world. For the islanders, it was not simply a tree. Its wood was carved into ritual figures of ancestral spirits, made into paddles, canoes, and sacred tablets inscribed with Rongorongo script. 56% of all objects from the island that reached European museums in the 19th century were made from Toromiro wood. The tree was literally everywhere, in every ceremony, every boat, every dwelling. Then the island was stripped bare. First by the islanders themselves, for fields and for the platforms that held the statues. Then livestock finished what remained. In the late 1880s, American traveler William Thompson recorded small groves of Edwardsia. The name Toromiro went by at the time, all dead, with bark stripped by flocks of sheep. One specimen outlasted everything. Protected by the crater walls of Rano Cow, it stood there for another 70 years, until the same man descended into the crater. Thor Heyerdahl arrived on Easter Island in 1955. 3 years earlier, he had crossed the Pacific on a balsa wood raft and become famous worldwide. Now he was looking for something else. Artifacts, traces of construction sites, evidence of how the islanders had moved the statues. Rano Cow, the largest of the island's 3 volcanic craters, filled with a freshwater marsh and ringed by sheer walls, was not a priority. But someone local showed him what was growing there. Heyerdahl described the moment in a letter to Swedish botanist Bjorn Alden, a longtime friend. He wrote of a small crippled and exhausted tree with one small branch remaining because everything else had been cut for wooden figurines. He knew it would not survive. "I collected a few seeds still hanging on the branch," he wrote. No ceremony, no photographs of the moment, no scientific protocol. He handed the seeds to paleobotanist Olaf Selling in Stockholm. Selling forwarded them to the Gothenburg Botanical Garden. Heyerdahl never returned to the subject. He had another battle to fight. His entire scientific life, he argued that Easter Island had been settled from South America, not Asia. Genetics eventually buried that theory. Heyerdahl lost the central argument of his life, and a few seeds in a Swedish greenhouse quietly germinated. In the autumn of 1958, Selling transferred 6 or 7 seeds to the Gothenburg Botanical Garden. 4 seedlings, all 4 from a single mother tree, almost certainly through self-pollination. This meant a genetic bottleneck. The entire species carried only one set of genes, but nobody was thinking much about genetics then. 4 seedlings in a greenhouse was already more than 0. Meanwhile, on the island, it was over. By some accounts, the tree was cut down for firewood around 1960. By others, 1962. That year, Karl Schanz, a German meteorologist working at the island's weather station, descended into the Rano Cow crater to see what he had read about in reports. He found an empty space between the rocks. No tree. The species was declared extinct in the wild. On the island, nothing. In Sweden, 4 seedlings in pots. The botanical garden made no announcement about this. The plants stood in pots among thousands of other specimens. The IUCN would not add the Toromiro to its lists until 1978, marked probably extinct. Until then, almost nothing was written about it. Gradually, descendants of the 4 seedlings spread to other botanical gardens, Bonn, Maintenant, Kew. In time, 2 more lines appeared. Seeds from Chile and a population in Melbourne, both without documentation of origin. Some plants turned out to be hybrids with related species. One specimen in a private collection in Chile was destroyed by its owner because too many tourists were coming to his garden to see it. Meanwhile, no tree grew where it should have grown. The Toromiro had been absent from Easter Island since 1960, and eventually someone decided to fix that. The first attempt, 1965. Seedlings survived for a few months, then died. The following year, again. Between 1966 and 1968, a secret planting was made that received almost no coverage, and again, nothing. Over 30 years, teams changed their technique, changed the season, searched for better locations on the island. The count reached 20 attempts. The result never changed. Then a different approach was tried. In 1988, laboratory tissue culture began. Not from seeds, but from cells under a microscope. Over 6 years, nearly 200 plants were grown. In 1995, 150 seedlings from Bonn and Gothenburg were packed in protective gel, transported across the ocean, and planted on the island. An international team, an official operation, photographs for the reports. Participants wrote that the reintroduction had succeeded. The next check showed otherwise. Not a single plant had survived, reached maturity, or produced seed. The reports of success had been premature. Estefany Diaz-Patti, head of the Conaf nursery on the island, described the process. Each seedling was transported coated in protective gel, handled with extreme delicacy due to its small size and sensitivity to environmental change. The trees could not withstand even the journey. Those that survived transport died in the soil within months. Nobody understood why. A quieter problem was emerging in parallel. It turned out that a significant portion of the plants in global collections were not quite Toromiro. Hybridization with related Sophora species had been happening unnoticed, year after year, while nobody was checking DNA. This meant some of the 20 reintroduction attempts might have been made with the wrong tree entirely. Until then, planting had been done blind. And while all these attempts continued, the Rano crater had held nothing between its rocks for a long time. There is a detail that does not make it into most publications. When 4 seedlings sprouted in Gothenburg in 1959, nobody knew the crater tree was still alive. There was no real-time communication between the island and Sweden. Chance did not descend into the crater until 1962, which means that for several years, the last wild tree in the crater and the first cultivated seedlings in the greenhouse existed simultaneously on the planet. 2 specimens of the species, separated by oceans, both alive, neither aware of the other. Then the wild tree disappeared. Only the greenhouse remained. While the official machinery kept failing, something else was happening on the island. Sonia Hawa Cardinali, a Rapa Nui archaeologist born on the island, became interested in the Toromiro through her own excavations. The tree appeared in the plant analysis of every dig she conducted, everywhere in the island's past and nowhere in its present. She planted 2 seedlings in her garden and began observing. No laboratory, no international team, no reports. Those 2 trees eventually produced seeds for the first time in decades on the island itself, 300 seeds. She called them mother plants. From their seeds, germination techniques were studied. From those techniques came the next wave of seedlings. From those seedlings, 31 seeds that she eventually gave to Attenborough. The answer to why the tree wouldn't take was found, and it was simple and merciless. For decades, scientists thought the problem was soil composition, drought, inadequate care after planting. They changed the technique, changed the season, changed the location on the island. 5 attempts by Gothenburg, 20 or more in total, and every time the same picture. Seedlings survive the first season, then die. The Toromiro, like all legumes, lives in symbiosis with bacteria in root nodules. These microorganisms, rhizobia of the genus Mesorhizobium, fix atmospheric nitrogen and deliver it to the tree in a usable form. Without them, the tree simply cannot grow normally in the island's depleted volcanic soil. The soil is low in nitrogen, and the tree cannot absorb it independently. Scientists from the University of Concepcion tested the soil of Easter Island and found no rhizobia of the needed type. The species had vanished with the tree. The bacterium, without its host, had followed. This is not a metaphor. When the last Toromiro stood in the Rano crater, its root nodules supported a colony of microorganisms that existed nowhere else on the island. The tree was cut down, and within a few seasons, the bacterium was gone too. More than 20 reintroduction attempts planted trees in soil where their partner no longer existed. Seedlings emerged from greenhouses where they had never lacked mineral nutrition into a world that held neither bacteria nor adequate soil nor moisture for them. The Gothenburg Botanical Garden participated in 5 of those attempts. 5 times, and not once did the species establish itself. When the answer was finally found, the reaction from the scientific community was roughly, "How did we not think of this sooner? Legumes, basic botany, rhizobium, first-year microbiology." But nobody put those 2 facts together and asked, does this bacterium actually exist on the island? Researcher Marissa Herding of the University of Concepcion described the moment when seedlings that had been growing for several months with inoculated bacteria were first inspected. The plants treated with selected bacteria had already become large trees, about 2 meters. They are much stronger, and we confirmed the presence of bacteria in their roots. 2 meters, twice as tall as any seedling had reached on the island across all previous attempts. Scientists are now searching for related Mesorhizobium strains on neighboring Pacific islands, bacteria close enough to the extinct strain to form a symbiosis with the Toromiro. In 2018, test plots were established on the island with seedlings pre-inoculated with bacteria from Chile and Isla Magdalena. The University of Concepcion is investigating microbial consortia, combinations of Mesorhizobium with other growth-promoting bacteria. It is painstaking, slow work. The Toromiro grows slowly, even under favorable conditions. November 2023, London, the Royal Botanic Gardens, Kew. Sonia Hawa Cardinali brought 31 Toromiro seeds from the island, from those same 2 mother trees in her garden. She handed them to David Attenborough in person. Attenborough, who was 97, passed them on to the Kew nursery team that handles exactly this kind of thing, species that no longer exist anywhere in the wild. Alex Barabalt, supervisor of the tropical nurseries, accepted the seeds and said, "Now I just have to grow them, no pressure." It was a joke. But behind it stood everything he didn't say aloud. Find the right bacterium, prepare the soil, persuade the island to make room for a tree it hasn't seen in 60 years, and do what 20 teams before him could not. 31 seeds. From plants that are themselves descendants of those 4 seedlings that sprouted in Gothenburg in 1959, from that one tree in the Ronokau crater, from that one branch from which Thor Heyerdahl took seeds in a year he didn't even note down. But there is a question that is rarely asked aloud. Even if Mesorhizobium is found and successfully introduced into the island's soil, which is not yet proven, the soil of Easter Island is no longer what it was. The island, 166 square kilometers in area, has over 8,000 residents and receives around 100,000 tourists each year. Tourism is the economic foundation. The native forest disappeared 500 years ago, and its ecosystem went with it. The Toromiro is not returning to a forest. It is returning to a cultural landscape where there is no place for it in any natural sense. Then there is genetics. All plants tracing their origin to the Gothenburg line, the majority of documented specimens in the world, are descendants of a single tree, most likely self-pollinated. The genetic diversity is catastrophically narrow. Some plants in world collections are probable hybrids. A 2021 study from the Universidad Mayor in Santiago developed molecular markers using the complete Toromiro chloroplast genome to distinguish the true species from crosses. The verification is not yet complete. How many of the 31 seeds in the Kew greenhouses are genuine Toromiro with documented descent from the crater tree is unknown. Barabalt knows this. No pressure is not a joke. Scientists now speak of 13 lines of Toromiro in cultivation, various clones and their descendants scattered among botanical gardens and private collections. But only one line is documented, the one maintained from Gothenburg, from Heyerdahl's seeds. All others carry questions about the authenticity of their origin. When the inhabitants of Easter Island carved the Toromiro into figurines, they were cutting down a tree that was keeping their soil alive. They didn't know that. Nobody in the 15th century knew about rhizobium, but when the tree disappeared, the nitrogen-fixing bacterium went with it. The soil became depleted. Without nitrogen, smaller harvests. Smaller harvests, less food. Less food, more conflict. The island that felled its forests to raise the grandeur of its statues eventually found itself in a crisis that some researchers consider one of the most dramatic ecological collapses to occur within a single human community. This is, of course, a simplification. Modern science, including the work of Hawa Cardinali herself, challenges the narrative that the islanders destroyed their own civilization. There is evidence that the ecological collapse coincided with other factors: drought, later contact with Europeans and introduced diseases, the slave trade of the 19th century. But the tree did disappear, and the bacterium disappeared with it. What they are trying to restore is not just the tree, but an invisible part of the ecosystem that nobody thought to protect, because nobody knew it existed. That is the true difficulty of reintroduction. Not the seeds, not the seedlings, not even money and will, but everything we cannot see and do not understand until it is gone. When researchers describe the future of the Toromiro, they tend to say, if the reintroduction succeeds, not when, if. The Toromiro is a slow-growing tree, prone to self-pollination, with minimal genetic diversity, without its soil partner, and without a forest to return to. The island has poor soil, an arid climate, and heavy tourist pressure. All of this is the reality each new seed enters. Right now, there are 3 fronts where something is actually happening. The first, the bacterium. The University of Concepcion, together with Murdoch University in Australia, are searching for Mesorhizobium strains on neighboring Pacific islands, bacteria close enough to the extinct strain to establish symbiosis with the Toromiro. The results have not yet been published as final, but these are the first attempts where the problem has been framed correctly. The second, genetics. The 2021 study from the Universidad Mayor in Santiago assembled the first complete chloroplast genome of the Toromiro and developed molecular markers to distinguish the true species from hybrids. For the first time in 60 years, it is possible to say precisely what is and is not Toromiro. Verification of collections around the world is ongoing. The third, the island. In the garden of Sonia Hawa Cardinali on Rapa Nui, living trees are growing. Not in a greenhouse on the other side of the planet, on the very island where the species disappeared. They do not yet form a population in any natural sense, but they are alive, and seeds are being collected from them. If all 3 fronts converge, with the right bacterium, verified seeds, and prepared soil, reintroduction may become possible for the first time in 60 years, not as a hope but as a technical problem to be solved. That if is still large, but for the first time in a long time, it has grown smaller,